One of the things we get asked a lot is, what does NeuroPRSM stand for? Neuroplasticity, Research and Support of Mental Health. And that acronym came about because we were all getting together and talking, fundamental scientists, clinical scientists, data scientists, and we realized that the question we were passionate about was, how does the brain change when mental illness arises, and how can brain plasticity be harnessed in support of helping the brain recover?
I love your term. You always insist on calling it co-facilitating. So we're not co-directing any sort of center here. We are just facilitating this incredible organic team that collaborates with each other in tremendously wonderful ways.
We're trying to understand more about how young people, early in the course of their psychotic illness, process information and how the neural representations of the dynamic world around them go awry. The goal is to identify where we might intervene or help the individual have a more adaptive interaction with their environment.
It's really that translational piece that I think about a lot and that I think NeuroPRSM really excels at. Doing translation is not always completely straightforward. Animal brains are not the same as human brains are not the same as animal brains. When we're manipulating circuits, when we're studying circuits, we're doing it in parallel ways in the humans and animals.
My interest in cognitive control really comes from trying to understand schizophrenia. It provided a model for understanding the relationship between behaviors that were important to functioning in the real world and the symptoms of schizophrenia, and then also the brain. In our translational work, we work as a team, and the more that team is able to communicate with itself, I can get ideas from the animal work that you guys are doing, and then also some of the tasks that I know that patients are having problems with, and then, well, let's see if we can do this in other model species so that we can gain those insights and begin to triangulate on where there might be some leverage for treatment here.
Our computational modeling of the neural level giving rise to the cognitive level really suggests some interesting changes in behavior and thought processing that may be going on with schizophrenia compared to healthy controls. For example, one interesting phenomena that we're starting to see is that we see changes in reaction times and how individuals are behaving based on presumably what this underlying cognitive process is that was able to be found because we were doing some of this underlying cognitive model to think about what the underlying process looks like.
I think one of the most compelling and unique parts of what we do is work on a computational model of decision-making that applies both to the kinds of animal models we use to try to understand the basic biology of psychiatric disease, as well as the patients that we treat in the clinic who are suffering from those diseases. And the power of that construct that we work on together is that it really enables us to translate in both directions, back and forth, from the animal lab to the human lab and even to the clinic.
I see the real strength in the modeling approach is to be able to ask in a computational way, how does this animal move through the world as a very small and controlled world, a decision world? How do they move through it differently than another animal with a different set of strengths and challenges? Ultimately, I see the goal of what we do with the animals to complement the human work, to both extend our understanding of how brains and minds of all kinds make decisions, but also specifically how, under challenge, brains and minds might make different kinds of decisions.
Computational tools help us understand individual differences in decision-making in mice. This informs us of potential brain circuits that contribute to neuropsychiatric disorders, so we can develop personalized treatments.
One of the really cool things about NeuroPRSM is that it causes a lot of those basic researchers to get out of our offices and go and actually be in the same room as clinicians and hear, oh wait, you care about that circuit? You want to know what this construct does? Well, hey, I've actually got a way to measure that. One of the great things about NeuroPRSM is if it brings us into contact with that computational language, and then that lets us line up these experiments across species way better. So now we can actually be saying, okay, what I'm doing over here is actually relevant to these people over here, and now we have a common language we can talk to each other in.
A lot of the work we're doing is with patients who don't respond to current interventions or have really severe illness who really need another treatment option. A lot of brain stimulation work is developed from more of this like bioengineering kind of physics perspective, and although a lot of good work has been done on TMS, for example, as a monotherapy, I think it takes these kinds of like disciplinary crosstalks to really think about like, okay, we're stimulating the brain, but we can also do a lot with therapy to induce brain states that might help people become more healthy. So I think that crosstalk has been really helpful in my thinking about how they synergize with each other.
For me, this is the future of discovery in psychiatry, psychiatric science. There is no way we're going to understand the complexity of these illnesses based on human studies alone, and there's no way you're going to understand the complexities of these illnesses based on fundamental science experiments alone.
I would actually go even further and say, I don't think we're going to understand the brain and even the functioning of neurotypical brain without seeing the range of how it can change under these psychiatric illnesses.